All right, well, welcome to the RMMS webinar, Advancing Clinical iPSCs for Regen Med, Part 1. My name is Josh Huntsberger. I'm Executive Director for Regenerative Medicine Manufacturing Society, looking to advance Regen Med manufacturing and workforce development through innovation and ecosystem building. In addition, I play a role in establishing collaborative frameworks to advance cell and tissue-based therapeutics through our working groups, perspective articles, and webinars such as these. My partner in crime, I don't know if she's joined us yet, but I'm going to introduce her anyway, Elisa Zilberman. She is overseeing key initiatives across RMMS working groups. She's one of our interns, where she's connecting scientific innovation with implementation. Currently, she's a grad student at Rensselaer Polytech Institute and is focusing on aging and microgravity effects on bone and muscle regeneration, as well as potential therapeutics. Before we introduce our speakers, I just wanted to give a little bit of background on RMMS and some activities. So RMMS is the world's first professional organization solely focused on advancing manufacturing solutions for regenerative medicine. It was founded to address unique challenges of scaling cell, tissue, and gene therapies, and it's bringing together thought leaders from industry, academia, government, and nonprofits to accelerate clinical translation of Regen Med technologies. We have 4 areas of concentration: cell and biomaterial manufacturing, standards and quality control, 3D bioprinting, and AI and automation. Everybody that's registered will have these slides, so you can access these links to join. There's a link there to join our working groups, and our next working group call is May 12th. I did want to talk about a few event opportunities on the horizon. So one is our Regenerative Medicine Essentials course public day. So this is the first time that we're doing a public day, and it's really looking at the future of regenerative medicine. We'll have a keynote from Dr. Anthony Atala, who's the director of Wake Forest Institute for Regenerative Medicine, as well as really exciting sessions in areas of AI, machine learning, virtual reality, in-space biomanufacturing. And that kicks off our Regen Med Essentials course. Essentially, that's a Regen Med 101 course. It's June 10th, 11th, and 12th in Winston-Salem, North Carolina. There's options to participate in person as well as virtually. And we do have a few limited opportunities for companies that would like sponsorship opportunities. Please contact Joan Schenk. She is our co-course director. And we're also working on another opportunity, which will be in the fall. This is our RegMIC conference, Regenerative Manufacturing Innovation Consortium. It's our industry-driven conference. So put that as a save the date, and more information will be forthcoming. I did also want to let folks know of some ecosystem opportunities. So we're currently recruiting companies, CDMOs, CMOs, raw material companies, shipping and logistic companies. You can contact our ecosystem navigator, Karen Barnes. We're also looking for veterinary regenerative medicine companies. So if you have interest, please contact Bert. We're growing an in-space biomanufacturing ecosystem, and our in-space lead is Jana Studemeyer. You're welcome to contact her. And then we also have training and workforce development opportunities. We're actually going to be launching a course in the fall on advanced biomanufacturing for all. Russ Reed can tell you a little bit more about those opportunities. There's also opportunities in the regulatory space where we have programs such as the Regulatory Navigator and our Clinical Trials Catalyst. There's also a test bed, which you can think of as a giant sandbox for advanced biomanufacturing equipment. Many of the folks that are on this webinar today have some of their equipment and products in the test bed. And just to highlight some of the things that are ongoing right now, there's collaborations with local universities to architect Regen Med training coursework. Cell Microsystems is demoing 2 organoid analysis platforms this month, and then the testbed is also looking to operationalize a fee-for-service contract model for companies that want their research conducted. And last but not least, there's also opportunities for companies to come to Winston-Salem and our region of service in the Piedmont region for office, lab, and biomanufacturing space. So with that, Oh, it says end slideshow. Great. I'm done, and I'm going to turn it over to our first speaker. And is Elisa on? Does she want to introduce, or otherwise I can?
Yeah, I'm here.
Oh, wonderful. Lisa, all you.
All right. Our first speaker is Steve Goldhood, who is the Senior Director of Commercial Operations at Pluripotent. Steve brings over 20 years of experience in chemical engineering and product development. At Pluripotent, he leads efforts in delivering turnkey iPSC solutions, including reprogramming, banking, and differentiation for both clinical and commercial applications. His work plays a key role in bridging R&D with scalable manufacturing. So he can begin.
Well, thank you for that kind introduction. Good morning from Seattle, Washington, where I'm based, and good afternoon to my colleagues over there in North Carolina and beyond. I'm going to take a little bit of time to share with you about induced pluripotent stem cells and how you might want to go about selecting those. So a few things that we'll talk about: stem cell opportunity, where you might find your sources and how you might reprogram those stem cells, some information about standardized cell banking, gene editing, and then some of the look in differentiation. So when I started my career 20 years ago, I was focused on medical devices. And as you think about cell therapies, these processes are nice. They can certainly solve some problems, but they're limited. Cost is certainly a concern. Resource availability, you don't have organs from every single person available all the time. The efficacy and immune rejection are issues. So as we think as moving forward, cell therapies are going to be maybe more applicable when we look at stem cells. And stem cells, of course, have the promise of being able to differentiate into any other cell type in the body. So we could then think that they'd be off the shelf, they'd be universal starting material, and we have the ability to sustainably supply those to the marketplace. So where might we get our stem cells? There's several sources. Of course, you can get embryonic stem cells. These have some of their own issues, but some of the nice things about them are they're very low passage, like less than 10 passages. They'd be polyclonal. They've been derived clearly from an embryo, so they can go into every different cell type. And some of the cells in that polyclonal pool might be more suitable for one cell type or another, depending on how far down the differentiation process or the maturation process these blastocysts have gone before that they've been isolated. And then there's the promise, since Yamanaka's factors, he won the Nobel Prize in 2012 for discovering these factors, that you can introduce to any cell type that still has a nucleus and get them to differentiate or to reprogram back into a stem cell. There's many ways of creating those. You can use integrative processes that would add DNA to the cell and transcribe these Yamanaka factors. You can use viruses to get there. It's just essentially the same idea: is put the proteins or the materials of choice into the cell so that they reprogram into a stem cell, and then they can be differentiated into any other kind of cell. You typically would find that they'd be clonal cells, and as the slide here indicates, it'd be closer to 20 passages or further advanced in age. And as stem cells, we'll get into this later, as they grow and grow and grow, genetic mutations can be introduced, and so we want to keep the passage numbers as low as possible. At Pluristics, the pluripotent stem cells that we can offer are kind of a mix of embryonic and the conventional iPSCs. Clearly, as you think about embryonics, the negatives are you've got ethical issues with the starting material. These blastocysts could have become humans, but the reprogramming method would just be to isolate the inner cell mass. The nice things about them are the polyclonality, I mentioned that earlier, and the low passage numbers. And the stem cells that Pluristics are able to make have those characteristics. The nice things about the conventional iPSCs, you've got an informed consented donor, so it's a human being that has given to research or given to the global effort their own cells and are happy to donate them, as opposed to taking a life or a potential life and turning those into stem cells. And then at Pluristics, we use a stem loop RNA. It's an RNA process, so it's non-integrative. The RNAs themselves are unmodified nucleotides, but the stem loop structure helps them stay stable inside of the cell. And so we end up with pluripotent stem cells that come out at a very low passage. That means they still have a lot of life left to them as they get expanded and then differentiated and turned into this cell therapy product. We've supported that with a drug master file. And another nice thing is you would only come to Pluristics to get a license to go into commercialization, as opposed to some of the other technologies where you need a technology license for the Sendai kit to do the reprogramming, and license the Yamanaka factors to use those factors for reprogramming. And then if you're doing gene editing, there'd be yet more licenses to get access to that technology. So at Pluristics, we can offer all of those in one package. Now, as we look at the banking workflow, what we would end up wanting to think about is kind of taking a seed train approach. So first you would make a bank of a seed bank, which you'd expand into a master cell bank, and then expand that into a working cell bank. And you do this with the mindset of a 30-year length of time for any clinical therapy. You want to have the same starting material on day 1 as well as day 30 or year 30 at the end of your lifetime for this clinical therapy. And so by making a master cell bank of, say, 200 vials, and each of those can make another 200 vials for your working cell bank, you end up with so many cells over the course of time that you're never going to need to get a new starting material. And that allows you to lock your process in and get something that consistently from the very beginning behaves the same way at the very end. And I wanted to also highlight that bank characterization of each of these stopping points is very essential. We've learned the hard way at Pluristics that if you have a genetic mutation in an early bank, it's not going to go away in a future bank. And so you could spend a lot of money making those future banks only to have to throw them out. And so what we do is we'll genetically characterize the cells at each of those stopping points very thoroughly. There's a couple places where pluripotent stem cells can get mutations. I'm not going to dwell on this too long, but you can get single nucleotide changes in the DNA structure, and you can also get changes of the karyoology. So one chromosome can get mutated and move to another chromosomal location. You get extra copies of chromosomes that would show up. So you need a range of assays, really, to see both the single nucleotide changes as well as the whole chromosome changes. And so at Pluristics, we'll do assays to see all of those things and keep banks that perform well and discard things that don't perform well. So coming back to this banking workflow, if you wanted to do a gene edit, which would be a very valuable thing to do when there's only a few cells present, you would want to do that kind of in the middle of this workflow. So after you've made a master cell bank, this is where you'd do some gene edits. And if we look at what kind of gene edits we might do, there certainly is the technologies available to do these gene edits based on CRISPR-Cas9, getting a license from the Broad Institute perhaps, or MAD7, a license from Inscripta, or there's other transposase opportunities to just change the genetic structure. These are important things to consider as you're thinking about your long-term cell therapy. And I want to highlight also that Pluristics can offer some platform gene edits. A few of those would be fail-safe. This is a safety switch gene edit. Essentially what it does is, as cells are dividing, if they are dividing, they're expressing a gene that we've put into the cell. And when you add a drug, that gene actually, or the protein that's been expressed by that gene, will turn the drug into a toxin. And so it'll kill cells that are dividing, but it will leave cells alone that aren't dividing. And the cells that aren't dividing would be your terminally differentiated cell therapy product. And so this is a way to remove a potential cancer that could form from your cell therapy. Other platform edits that we have are immune evasion types edits. We can knock out the B2M and the C2TA genes. This would be the gene that's responsible for holding the HLA1 or the HLA2 markers on this outside surface of the cell. And then this way, it essentially makes it look like it's a normal cell in your body. And then another opportunity we have for immune evasion is this IACT stealth cells. This is another technology that Pluristics has licensed in exclusively and makes available to our customers. But it's an overexpression of different factors that actually convince the T cells or the other B cells in your body that the cell is actually host cell as opposed to foreign. So those are two key ones, right? You can make the cell look like it's part of the body, and you can also have a safety switch to kill the cell if they go rogue. So I wanted to transition quickly and talk a little bit about differentiation. This is the point where I think most of the value from stem cells actually needs to be spent, the time spent to develop that value. And what we hear from labs is, hey, I've got this cell line, it's my favorite cell line, but one day it stopped differentiating, or I'm genetically modified and it no longer differentiates the way I want it to, or I've got this new cell line that I'm trying out, but it doesn't work like the last cell line I had, so what's going on? And what we see many people kind of come up with, the normal response is, hey, let's throw everything out, let's buy fresh reagents, let's run the differentiation, and that should solve everything, right? Or maybe I'll re-optimize the culture conditions, or I'll just try a different cell line. And nobody really stops and thinks, why did it stop working to begin with? And what I'd like to propose and give a little bit of guidance on here is that some standardized differentiation workflows can be very helpful when you're establishing your processes. Stem cells, I have learned, are very finicky. They like to be fed on a very routine schedule once a day at the same time frame. And so if you build your differentiation process with that in mind and condition your cells to be having not only feedings on a regular schedule, but also defined passages, so you're not passaging willy-nilly, oh, let's passage today because it's the weekend coming up, but just continually go with the same processes. That usually really helps the cells behave routinely from the next time to the next time that you're doing the same differentiation. And you want to make sure you're using the same media, the same matrices, the same cryopreservation methodologies, so that you're getting a very uniform process. It's really that consistent differentiation requires a stable and reproducible process. A great analogy to think of here is, and this is, personally true for me, when you're traveling around the world, it takes a little while to return, to kind of get your body back on the right clock from jet lag. Yesterday, my son and I flew back from Florida to Seattle, finished our spring break, and this morning at 5 a.m. my body woke up and said, I'm hungry. Well, it was 8 o'clock on the East Coast. And in the same manner, the cells that you're working with and differentiating are conditioned to expect these processes to happen at the same time. So you don't want to start the differentiation early or late and expect the same results to happen from one run to the next run. So some results that I'd love to share with you guys, just to show the potential of pluripotent stem cells and their differentiation process, is some work that was done by our partner Defined Biosciences with some grant money from the California Institute for Reproductive Medicine. They took some of our cell lines at Pluristics and grew them for 10 passages and then started their standard differentiation protocols. And these are all protocols that are known protocols. They were able to show that all these different cell lines could go into these different fates, epithelial, neuronal, and they were looking at, the cell surface markers. I don't expect you to actually see the details of all these flow plots, but you can identify that they were able to make the right kind of cell types, and they did a full genomic panel using stem genomics, where this is a great assay. They look at different hotspots in the iPSC genome and find spaces where there's either amplification or reduction in chromosome number or in copy number. And saw very stable results with their cells. But I think really the fun one here is the functional piece, right? So they could also show that these cardiomyocytes that they made were beating, they were moving around, and that for me is the satisfying part of stem cell biology, is watching the cells actually move around in the petri dishes. So a possibility. You can certainly do all the differentiation. And so with that, I do want to give some time for my co-speakers to share more details about automation and the right kind of environments. These are also key pieces of the stem cell story, and I'm not going to steal their thunder, but I'll give them the space to do that as well. So thank you for the time and the attention, and I look forward to learning along with you guys more about what else is available.
Wonderful, great talk. And if everybody can hold your questions till the end, we will have time for discussions. And Elisa, do you want to introduce the next speaker, please?
Sure. So next we'll hear from Lynn Frick, who is the CEO of CellX Technologies. Lynn leads her team in advancing the automation of cell therapy manufacturing. Her work focuses on integrating robotics, imaging, and AI to streamline GMP production and address key bottlenecks in iPSCs-based therapies. She brings a strong multidisciplinary perspective to the challenges of scalable manufacturing.
Thank you. Hi, everyone. Well, I'm here to speak with you today about our automation platform and its applicability to advanced clinical iPSCs. Next slide. Just a bit about CellX. We are a Cleveland Clinic spinout, and we're located in the Boston area now. Our mission is really to streamline cellular therapeutic product development by applying automated development and responsive automation in the production of all stem cell therapies. So iPSC is just a subset of what we do, but it's the name of the seminar, so we'll go with that. We want to talk about what problem we're solving. I want to tell you a little bit about the Celligent system and our platform solution, and then I'm going to take you quickly through case studies that you can always question us more on, and we're happy to get back to you, and a little bit on our early stage translational services. Next slide. So I think Steve already hit on the problems, and I think everybody knows that stem cells doing this the old-fashioned way, very manually, is just a long, labor-intensive process. Even in our hands, when we're getting the automation set up, everybody's doing things manually, and there's a lot of late nights and weekends and frustration. Even in people who know what they're doing, it doesn't always go smoothly. So really, our problem is, how do we get some subjectivity out of the process? How can we make this more consistent? And I think that Steve kind of led into this, where if the process can be consistent, you can get rid of just a lot of the variability. And then, from human to human, there just is high variability. People do this as an art. They get really good at it. You have the one person who's extremely good at it, and everybody else looks bad relative to them. So it's a problem. Poor scalability, also a problem. Just how do you scale this out? And then, of course, the paperwork side of this, I actually was in a GMP lab where they were doing a phase 1 material. Somebody was working in the hood, somebody's behind them with the iPad trying to fill in what they were doing. And they had this whole discussion about, I'm tapping the plate. And the woman behind with the iPad was saying, how hard are you tapping the plate? I don't know, I'm doing 3 taps. So these are the things that lead to inconsistent processes when you're going into GMP. Next slide, please. So the questions that drive us as a company is, how can we put quality into the process? So how can we drive toward process control? Process control with a living product has all kinds of challenges, but this is what we want to drive toward. And we do that by building reliability in via this automated repeatable protocol and data acquisition parameters, algorithms, and data annotation that are consistent. How can we take automation to reduce this clinical risk? Well, we could do a couple of things. We can automate positive and negative cell selection. We can look at image consistency, and we can handle all the liquids for the media and the feeding, as we spoke about before, and the plate handling, and make those more reproducible. And then the one thing we're finding is if you want to really apply AI beyond machine learning, it takes a very consistent data set. So how can we replace manual processes and decisions in an application agnostic way, but also how can we collect the data and put it in a structure that makes it accessible to not only machine learning algorithms, but the next stage of real AI and knowledge graphs, et cetera. Next slide. So what we've come up with is what we call the Celligent. It's short for intelligent cell processing. Really, the idea was to purpose build an instrument that combines image-based analytics so that we can have the visual record over time. Think about it. Every day you're taking that image, you have the record from the time that you reprogram, and you are creating a way to automatically and passively create documentation. Also, by applying automation, we are mimicking the manual process. We are not trying to do anything fancy. We are doing what the human beings are doing, but we're doing it consistently and with no variation. We're able to operate 24/7. And this robot has surgical precision. This is not a liquid handler. It's a little bit more precise. And you'll see in a minute that allows for some precise picking and weeding. And then again, we're trying to have a process that allows a subject matter expert to train an algorithm. And at first, it's a subject matter expert who's responsible for saying, these are the right cells, these are not, based on going through the whole process, looking at the analytical data, going back through the process, looking at those images, training through image segmentation, what that looks like, and then having that algorithm get better and better as time goes. Next slide. So just to tell you, so you could look at it, what it looks like, what it does. The top view looking down is the top of the Celligent deck. There's a donor plate and a recipient plate. I'm just showing the 6-well format. It can be any kind of plates going in here. This is just for simplicity throughout. I'll keep it consistent. The image on the right is the actual aspiration tip going down to pick cells in this place. And in the bottom, you'll see the Celligent in an environmental chamber integrated with a robot. And that is the biospherics chamber that Alicia will be speaking about, so I won't take her thunder. But just so you know, if you go to the next slide, I have a bigger picture. This is the one at the Cleveland Clinic, and it consists of 3 modules, a laminar flow hood on the right, a GMP pass-through, and then what we're calling the processing chamber. And if you look at the photo to the left, it's the head-on picture. By integrating it into this environment, it really gives you a lot of modularity and flexibility into how you configure things. We are also configured with a automated incubator. If you look to the far left of this picture, there's a shuttle or a tunnel that is under the same conditions that shuttles over to an automated incubator. There's barcode reading. Everything that comes in here is tracked, traced, and documented in that GMP compliance software. Next one. So I'm going to do a working example, very simplistic, but just bear with me just to show you kind of how this works and what parts of the process we hit. And for this, fibroblast to iPSC, just as an example. We can also look at the differentiation part, but this is just kind of the most simplistic view. And if you look from the time you transduce the fibroblast all the way through passage and expansion, multiple passages and expansions, there's a lot of work that goes on in 6-well plates. And everybody's been in the lab where they said, oh, I want you to do 3 of those cell lines. I want you to do that in triplicate. And all of a sudden, the number of plates on the other end, oh, whatever, that's 140 plates now, right? So how do we take this and try to automate it? I'm just going to walk you through some of the possibilities. Next slide. So the first thing we can do is we can monitor and feed cultures. So we could document the reprogramming phase and actually look over time. And if you just look day 6 to day 26, you get that image every day and you're seeing what's going on. And we can also do a biopsy where we very precisely remove a sample. That sample goes out for RNA-seq, karyotyping, whatever, whole genome sequencing. That information, however, comes back into that database as annotated at that time point. So now you can imagine if you biopsy every day or every 3rd day or whatever, you also get this record of exactly what's happening over time that you could look at after the fact or retrospectively, and you can actually mine that data with certain algorithms, and that's where the AI part starts to come into this. Next slide. So you can analyze emerging colonies, and just to point this out, our colonized software allows the user to identify clones that meet their CQAs. If you look at the green circles on the right, that's C-suite finding the colonies on the dish. The SME is the subject matter expert, the yellow rings, and you'll see the actual C-suite identified a couple of colonies that the subject matter expert missed. This happens all the time. Once the algorithm knows the morphology, it scans that large field of view image on the left very, very quickly and goes through and finds those colonies. So it's a way where you can, again, to Steve's word, add consistency, and that you're establishing and you're meeting or exceeding these defined metrics. You can also have exclusionary factors like, I don't want to pick a colony that's too close to another one. So there are ways to rank in addition to just looking for the colonies. Next slide, please. We can also weed. So we can precisely remove fibroblasts and poor quality cells, and it's incredibly precise. So you can do it even easier than I'd say. You know, a lot of people who are very good with a pipette can do it well, but then there's always this training time of getting people to do this well. This can do it off the bat. And if you look on the left, before weeding, you can see the area around the colony is all has cells. On the right, it's post weeding. So in this way, you can actually select with a mouse kind of weeding movements, and then later you can create an algorithm to automate that weeding. And I'll show you that in one of the case studies. Next slide. You can also identify desirable clones. So the visual assessment of the image or colonized software can identify these clones, and then you can take out areas of spontaneous differentiation, or sometimes when the middle of the colony gets too thick, you can actually just take out the very center or the donut part of the colony and donut hole part and remove that to make sure that only the highest quality cells are maintained. Next one, please. The other thing we do quite well is clone picking, and this is one of the fundamental patents that we've exclusively licensed from the Cleveland Clinic, and that's atraumatic picking, where we are hovering over the cells, aspirating them. The nice thing about it is the clumps are of similar size to what you would do manually, and this is a bit of a trick of the automation. It takes a while to get there. You can optimize the process to create the right size clumping, and that is when you deposit the cells, they readhere quickly and start growing again. And so in this way, you can automate the passaging procedures, and we can do what's called a pick passage, where we're picking very specific parts and passaging. Now we can also do bulk passaging, where we're passaging the whole plate, swirling, tilting, everything you could do manually. But pick passaging sometimes is a way to decrease the number of passages and not needing to go to passage 10 anymore. Hey, I could get it done in 6 passages. So it cuts time. It can make a high quality culture. Next slide. Just a word about the Celligent GMP platform. This is being built right now. It'll be delivered in the 3rd quarter for GMP production. So I'm showing the schematic again of the biospherics. There's also a control cart and 2 automated incubators in this particular setup. And we had to do some engineering to make this applicable to GMP from what was kind of a process development system that could go in any biosafety cabinet. And that was, you know, we wanted to regulate the temperature of the media, for instance. We wanted to have tip and waste below the processing chamber. We wanted to create a vacuum so there was no aerosolization within the chamber. So there's a bunch of modifications that have been done. A new single-use kit that allows operators to change over the entire product contacting easily. All that is being implemented in this GMP software, which will be shipping in 3rd quarter of this year. Next one. So just 3 quick studies. One is an autologous photoreceptor replacement. The other is an allogeneic cardiomyocyte CQA assessment. And the third, an autologous cell therapy. Just going to do a quick 2 slides on each to give you a flavor. Next slide, please. So the first one, I won't go into detail because I think Alicia probably has more on this, but Bud Tucker at the University of Iowa, you know, originally was one of our first customers, and he worked out really the production of these clinical grade patient-specific iPSCs. There's a paper in the Journal of Translational Medicine that I'm glad to get you a copy of. Next slide. And the one I like is this precise picking to leading to better clones. This is from his paper, The Picking and Clonal Expansion. If you look at the middle panel, those are the wanted or the best iPSCs, and the panel on the right is post-picking. And look how cleanly it was able to remove and put those ahead. So really this project included fibroblast iPSC generation, clonal expression, then retinal differentiation to create the retinal organoids. Next slide. The next project is one we did with the Advanced Regenerative Medicine Institute, not to be confused with this group. This is one of the Manufacturing USA 6 sites that was set up for tissue fabrication called BioFab. It's here in Manchester, New Hampshire. We wanted to look at, in cardiomyocytes, if you have 3 different cell lines, how can you actually determine CQAs perhaps easier, and how can you maybe improve the passaging through selective colony picking? So if you look on the top slide, the green circles are kind of pick points. The slide below is the picks post-pick, so the cells are gone. We actually were doing this to allow for specific colony expansion to enhance the line's cleanliness and to improve the differentiation efficiency. And this was only really feasible through automation. If you go to the next slide, I'll show you what I mean. So when you look at the results of the positive picks and the negative picks, if you took the positive picks, look over to the right, the negative pick, the middle panel, the manual kind of bulk passage comparison at the bottom, actually the one with the positive picks is just a cleaner, nicer culture, and it was done more quickly. So this is kind of the promise of that you don't really know you can do it better until you apply the automation. Next slide, please. And one other one I wanted to speak to is this is a customer, Aspen Neuroscience. This has been publicly presented at the Hanson and Wade Manufacturing Conference in 2024 by Will Ansari. He's the associate director of tech dev for Aspen, and they are making an autologous cell therapy composed of a dopaminergic neuronal precursor cells, and it's to replace lost dopamine-produced neurons in the brains of people with Parkinson's disease. So Aspen has run a lot. They've spent a lot of time on the platform. As you can see, they also have it in a biospherics chamber. It's been a very good project. They've done a lot of time, with the operation, 17 months of testing the device, and it's worked very well. Next slide. And I just wanted to take another. This is again from Will's publicly presented presentation. The cleaning paths are autonomously generated. So they've created an algorithm, and they're specific to their cell culture that does this kind of weeding. So when Colonize goes and looks at what's on the plate, it'll actually just start automatically weeding around everything that needs to be weeded. So in this way, they are providing for autonomous production throughout their process. Next slide. And just a word on some of our services. So we understand that each process is unique. There's a lot of challenge in stem cells. Some of the things we're doing is helping with the translation to GMP. I almost call it a pre-CDMO. We're not trying to be a service company uniquely, but we want to help our customers and enable them with CQAs or critical process parameters, perhaps de-risking the regulatory process. And we're set up that we can work with very early stage companies. So it's kind of a nice compatible thing. Next slide. And some of the ways that we work together is with a Celligent system or platform installation, but then we provide the support. We have broader licensing and collaboration deals, and we have targeted services, again, to support this very early, almost R&D to process development to early GMP. Some of the stuff that needs to be worked out, perhaps scope before going to a bigger CDMO. And then we have a shared program management, which is kind of in step in a milestone-driven process relative to the company's funding, which we all know has been tough these days. There's been a lot of starts and stops, and so we're sensitive to that. We try to find a way to work with people to help them out. So with that, my email's on the last slide. If anybody needs anything or would like to see more, please feel free to reach out and contact me.
Wonderful talk, Lynn. I put in the chat some ideas for you to think about, and we can definitely follow up.
Okay.
Keeping on time, because I want to make sure all our speakers get sufficient, and we will hopefully have a few minutes too for discussions. Elisa, can you introduce our last speaker, please?
Yep. So our final speaker is Dr. Alicia Henn, Chief Scientific Officer at Biospherics. Dr. Henn is an expert in controlled manufacturing environments and physiological cell processing. At Biospherics, she is driving innovations that support scalable, contamination-free iPSC production through environmental control and closed systems technology.
Thank you. Thank you for that nice introduction. I am going to start today, if I can get my slides to shift. There we go. I'm going to start by talking about the environment in which cells are traditionally produced for clinical use. Clean rooms, traditional clean rooms, typically take many years to plan and build. By the time they're built, the processes that they were intended for may have changed because it's very difficult to plan years in advance of what you need. They're restricted in use in that once you have a clean room, it's always a clean room. You can't really use it for other things very easily. Operators tend to leave them in the dark to try to reduce the huge HVAC costs, energy costs it takes to run these, unless they have a process that's going in it that can pay for that energy use. And workforce turnover is tremendous because people hate working in full clean room garb. It not only takes a lot of time to climb in and out of it, but also it's quite expensive. So, and even with the high HVAC costs and all the garbing and cleaning, cells still face contamination risks, even in clean rooms, mostly from people in the room. We're the source of contamination for them. The worst part of it is that no matter how clean it is in that room, no matter how hard we work to keep it clean in that clean room, it has the same conditions for cells as the crudest, rudest laboratory anywhere in the world, because it fundamentally isn't designed for the needs of iPSC or any type of cells, really. Room air is fundamentally hostile, non-physiologic, and highly variable environment for cells from inside the body. Now, in contrast with that, we designed the ex vivo system, which Lynn's already talked a bit about, to provide an environment that's safe for cells. Compared to clean rooms, it has reduced operations and maintenance costs. It's modular, it's scalable, it's movable, it's much more flexible to use and to change over time. It's much more operator friendly in that people don't have to wear the full suit garb for clean rooms, and it has that separation between the cell space and the people space, which automatically mitigates a lot of the microbial risk that cells still face even in clean rooms. And it's fundamentally cytocentric. It's designed to be cytocentric. And what do we mean by this? What we mean is that it's an environment that meets the needs of cells. It's designed specifically for the needs of cells. So it's closed and controlled so that the entire process is protected from start to finish in a controlled environment. And with this controlled environment, you can create optimal conditions for iPSC. Ones include things like physiologically relevant oxygen levels, which I'm going to come back to a little bit in some of the cases I talk about here, as well as constant CO2, constant 37 degrees. And there's no exposure to the variability that you get even in a clean room environment when cells are exposed to room air. So with the ex vivo system, it mitigates microbial risks in a couple of unique ways. First of all, the cells are physically separated from room air, and the personnel that are there that produce a lot of the microbial risks that cells are exposed to outside in room air. There's constant HEPA filtration of the atmosphere inside the system. And most importantly, the atmosphere inside comes from compressed dry gases, dry gas tanks. We actually have a patent on the process that we use for controlling microbial risks through using controlled humidity in cell handling areas so that microbes can't grow there. This in turn allows operators to dramatically reduce the use of antibiotics and biocides that their cells are exposed to, which can be particularly important for iPSC. As I said, it's modular, it's scalable, it's movable, which means that it can adapt to changing protocols. And we have customers that often want to bring in automation. They want to scale up, they want to scale out, they want to change their process, they want to make their IPS into new products. It's easy to accommodate these changes because our system is so modular. We have customers like Bud Tucker at University of Iowa that will call us up and say, hey, I've got some new automation. I've got this really cool cell picker I want to put in my system. Can you make a module for it? And of course we do. And we can integrate it into the rest of this process quite easily. So you can take this automation for iPSC production, like Lynn was saying, and you can now take the reproducibility of those actions and the recordability of those actions and combine it with the controlled conditions of the environment. So now you have much more predictable, better constructs with iPSC, because of course we know the uncertainty is the bane of any business, especially for one generating tissues from patient cells, which are a highly diverse source. So the ability to control and clone an environment and put it anywhere combines really well with automated cell processing. So now you can have those reproducible conditions as well as reproducible operations. And if you combine that with a reproducible cell bank, now you've got a better cell product in the end. The ex vivo also removes any particles that are generated by moving equipment, which helps mitigate any particle risks that automation might add to the cell environment. Trailhead Bio, which uses a system on the right, which encloses a huge TCAN pipetting robot, has said that one huge benefit that they got from the ex vivo was that once their automation was disinfected, the ex vivo kept the equipment far cleaner than it would have been outside. And this saved them a tremendous amount of time in disinfecting their equipment over and over. Now, this is the ex vivo system in the WFirm test bed. And this is one of my favorite systems, I must say. Here, cells can stay in fully optimized conditions without having to be exposed to variable room air for things like trips to the centrifuge, trips to the microscope, or other equipment. And I'm going to show you on this system some of the anatomy of the ex vivo that makes it work well, some of the different modules that we can combine to make a custom process. Behind the computer here on the left, we have a laminar flow hood. That grid in the back is a HEPA filter, and air is blown through the HEPA filter and across this workspace, making a clean space that's used just for staging and surface disinfecting items before they're imported into the ex vivo. These buffer modules that you see with the doors on it in the middle, these are for changing over the gases that are around imported materials to that filtered tank gases that I was telling you about. And then the modules after that are for processing and incubating cells, and they can contain all kinds of needed equipment, like the centrifuge you see here. There's another module that's behind there with incubators and microscopes. There's a chill chamber on it for cold conditions if people need it, as well as 37-degree control that's possible in there. It's a great system to try out a potential iPSC production process. So we have customers that are using human iPSC and have published on these in numerous places. Just to touch on a few of these, we have customers that are producing cord blood endothelial cells from human iPSC, retinal endothelial cells, cardiac organoids, neurons. And this paper I particularly like in that this is Mana Paras group, she was generating trophoblast from human iPSC and using them to model preeclampsia in their lab. Also, we have other customers like the Phil Schwartz group that's generating human neural cells from iPSC. And he recently published that they've shown that they can take these human neuronal stem cells and inject them into mice, and they migrate through the blood-brain barrier, distribute throughout the brain, differentiate into glial cells and are able to partially restore the enzyme deficiency that otherwise would lead to mucopolysaccharidosis type 1 in these mice, also called Hurler disease in humans. And also on the paper on the left, I mentioned this just because they published years ago in the Journal of Visualized Experiments, actually some videos on how they handle their stem cells in the ex vivo system, which is kind of cool. If you're interested in those videos, let me know. So, and this is the system that Lynn was talking about in Bud Tucker's group in Iowa. They're growing iPSC and, of course, using CellX automation in the ex vivo. One of the things I wanted to point out about this is that they use the ex vivo system to control oxygen so that they can use oxygen as the signaling molecule that it is. So they use low oxygen to generate their iPSC. And they say in this paper that it's particularly helpful for reprogramming somatic cells from elderly people because it's so difficult sometimes to get clones from those cells. And then they use high oxygen then to differentiate those into retinal progenitors. So using these controlled environments, you can actually drive iPSC behavior. They have a very recent preprint. This isn't peer reviewed yet, but I can't wait till it is, because they talk here about how they've found that 5% oxygen is optimal for their iPSC clone generation. And they found higher oxygen is really great for generating retinal organoids. They've got a system now that's using all xeno-free reagents, and they were able to take these retinal organoids and put them behind the retinas in rat eyes and actually see them form new synaptic connections with host neurons. So this is incredibly hopeful that we will see something like this soon in a human clinical trial as well. And this is all under cGMP clinical grade cell production conditions. So I want to talk for a minute about the 3 L's of iPSC production, and that's location, location, location. There's another paper out from Bud Tucker's group where they talk about some of the difficulties that those that are generating iPSC for clinical use are really coming up against. And one of the things, of course, is that you need cGMP conditions to generate, maintain, differentiate, and store these cells and constructs until they get to the patient. So using offsite CROs can be problematic in that now you have shipping time. So now you might need to cryopreserve those. And some of these constructs with neurons are too delicate to survive that. So having production placed near surgical sites and near the patients is actually something that can make a huge difference to their processes and to getting these cells to the clinic. So, of course, with the ex vivo system, it's perfectly designed for a decentralized manufacturing for iPSC for clinical trials. You can lock in the conditions that you need for optimal iPSC growth, whether it's oxygen levels, CO2, temperatures, you name it. You can lock those conditions into your process and also with the same modules, and then you can clone that system and put it anywhere in the world or in space. And you can monitor and even control those conditions from a central location, which gives you better efficiency of operations and better central control of what could be a very nice decentralized manufacturing format for iPSC generation for the clinic. And that way you can have your live cells, your live constructs near where the patients need it. You don't have to cryopreserve and ship. I'd also like to mention this one case that we had that's really special. It's close to our hearts. It was a FDA-approved compassionate use case for a little girl, a toddler, who needed a new trachea. And they needed a place to produce this trachea in Peoria, Illinois, and they didn't have it. They were going to have to fly her cells and tissues back and forth to Chicago to the closest clean room. So we took an ex vivo system right out of our showroom and transported it to the hospital there and set it up, had everything disinfected, validated. We had the team trained and everything ready to go for generating this trachea in 6 weeks, which is a huge benefit of this modular system. And we have ex vivo systems all over the world right now and used for producing clinical grade cells, not only iPSC in Ottawa, but also pancreatic islets in Melbourne and now Adelaide, Australia, cardiac progenitors in Ottawa, CAR-T in Singapore, MSC in New York City, all kinds of different cell types in all sorts of different places. So I'm going to stop here again with my ex vivo system, my favorite one, and invite you to come try out your iPSC-based processes, including oxygen control for your iPSC. Contact me or Josh for more details on getting into the ex vivo at the test bed.
Awesome, awesome. Well, you guys did a wonderful job. All your presentations. Alicia, I left you a little note in the chat on just how much I enjoy your platform and where I think it is leading as towards a future platform for biomanufacturing. I'd love to have maybe 2 discussion questions if we could. Because I think this group is phenomenal. So it'll extend past the hour. Is that okay if we spend about 5-10 minutes past the hour? Okay, great. So one of my questions is, what do you see as the most critical bottleneck today in translating iPSC platforms into clinical grade therapies, and how is your organization addressing it?
I could field that one.
Yeah, go for it,
Yeah,
Steve.
It's a great question, Josh. There's 2 things that come straight to mind right away. One is the desire, the human desire to build everything from the ground up rather than buying some starting material. And then the second thing is to keep a clinical mindset. So you're choosing the right materials, the right location, the right technologies that are going to work when you move into clinical. So at Pluristics, one of the things we believe is supplying this consistent and well-characterized clinical material. And that way, our partners, you guys, everyone who's benefiting from these pluripotent stem cells can focus on doing your own differentiation, making your own cell therapy, and moving your process forward. And you don't have to think about the raw material. So.
Yep. Great. Lynn, you're on mute.
Take a little different approach in that we think that sometimes producing the raw material leads to a consistent and maybe better differentiation, and there's probably evidence for both ways depending on the situation, but it gives an optionality through automation that would be hard to do in multiple cell lines without automation. Yeah.
Mm-hmm.
Yeah.
I'd agree with all of those and add to that, clean room space, even if you're looking for it, is really difficult to come by, particularly since the pandemic. And so having iPSC production environments anywhere you need them is something that can really help unlock that.
Awesome. So my next question, how are automation and AI shifting the regulatory and quality control landscape for iPSC manufacturing? And how are these advances contributing to more robust iPSC manufacturing processes? Maybe Lynn, you want to start off, because
I think
talked about that, and we talked about that a little bit yesterday too.
We did, yeah. In cell, just recognizing the phenotype, morphologically. There's been a lot of research done in the diagnostic area with radiomics, et cetera, that has kind of led the way in some of what's going on now, or just starting to go on in stem cells. And we actually feel that some people will say, oh, morphology isn't enough of an indicator. But once you collect the data and biopsy it and bring all that into one database and can actually learn about it in a more holistic way, then you can go back and the algorithms will find things that you couldn't even see with your eye. So I think the potential is tremendous, and that ultimately it may lead to a time, and I think this is regulatory wise in the future, where we can actually have prediction and adjustment to our processes, because these are living processes as we go. So that kind of well, Steve, to quote the chemical engineer, feed forward control, right? Which is kind of the dream of every manufacturing process. It would be very hard to do today and how we're measuring things, because it's hard to measure things. But if you could come up with a way to do that reproducibly, it could be really, really a way to change things.
Yeah. And Alicia, this one's for you. What role do standardization and modular platforms play in accelerating commercialization for iPSC-based regenerative products?
Well, I see huge potential in decentralized manufacturing. I mentioned it in the talk, but I think that this has the ability to really solve some of the biologically founded restrictions that we have on things like shipping. And so I think that decentralized manufacturing also fits really well with AI and centralized control, and sort of having, I'm just going to come back to your other question here too, and just say, using AI to control even the environment with feedback from the cells, so that you can drive real-time changes in the environment, are ways that you can use a combined environment along with some of these new exciting AI and location-based technologies to control from a central place, as well as let the cells tell you what they need in the moment for a process.
Yeah, great. And then this one is really for you, Steve, but others can jump in. As we look towards allogeneic iPSC therapies, what considerations should developers prioritize when choosing starting cell lines for broad patient compatibility and safety? And maybe talk about some of the tools that you guys have in place.
Yeah, I think you hit it there. It's the patient compatibility and the safety. So with Pluristics, we've got both the opportunity to use this fail-safe gene construct, which is the kill switch that would keep your cells safe, and then the universal cells with the immune evading technologies, either the B2M, C2TA knockouts, so get rid of your HLA markers, or overexpress these other factors under this IACT, induced allogeneic cell tolerance technology. And then that way, the cells that would ordinarily be looking at as foreign by your body would be seen as natural, and would persist then in the body without taking immune rejection drugs for the rest of your life. So those would be what I'd get.
Great. Well, I think I'm going to stop it there because we are past the hour, but I do want to thank all of our distinguished speakers. Really enjoyed your talks today. Fabulous. You guys are doing tremendous work for the field, really innovating, and thank you very, very much.